venous systems, and end with the cranial nerves and the short quiz to test your memory. Fine. I'm going to start now with the cranial vault. So the cranial vault is actually the space that encompasses the brain and protects the brain along with the base of the skull. I'll talk today about the calvaria, and in 2 weeks you'll hear more about the base of the skull. And the calvaria is constituted of the following bones. We have the frontal bones, we have the parietal bones, we have the squamous part of the temporal bones, and we have the interparietal part of the occipital bones. And these bones are held together by sutures. And the main ones I want you to know about are the coronal suture, highlighted here in red, which joins the frontal bones with the parietal bone. We have here in green the sagittal suture, which is in the midline, joining the two parietal bones together. We have here in red the squamosal suture, which joins the squamous part of the temporal bone with the parietal part. And we have the lambdoid suture in the back, which joins the occipital bone with the parietal bones. Let's look at those in more detail, starting with the frontal bone. So we can see the frontal bone anteriorly over here with the coronal suture. And isolating the frontal bone, we can see how the coronal suture goes around like a crown, hence its name. And on a sagittal section of a CT on a bone window, we can see the zigzag line over there, which refers to the coronal suture, and the bone anterior to it, that's the frontal bone. All right, let's move on to the parietal bone, and isolating it, we can see how it's related to all the other sutures we have discussed. And on an axial slice, we can see here as well, anteriorly we have the right frontal bone, the zigzag line on either side, that's a coronal suture, so it's a continuous suture. You don't have to specify right from left for this one. You have the right parietal bone, which is just posterior to the coronal suture, and here we have in the midline the sagittal suture. We are looking at it from a top view, which is why it's posterior over here. Next, you have the temporal bone, and the temporal bone is isolated by the squamous suture over here, or squamosal suture. So we can see the sagittal suture again in the midline. We have the right parietal bone, and there's this faint suture that you're seeing on either side, which is oblique in shape, and that is the right squamosal suture. And just inferior to it, we have the squamous part of the right temporal bone. Finally, we have the occipital bone, which is isolated from the rest through the lambdoid suture. And on axial CT, we can see the occipital bone in the back, and the zigzag lines on either side. This is a lambdoid suture. There is one more suture that is not seen in everybody, and that is between the frontal bones. So, as children, most of us have this suture. However, when we grow old, it will fuse and um, disappear. If it persists for some reason, then we call this a persistent metopic suture, and that is a normal anatomical variant that could come up in the exam. So it's worth knowing what it looks like. And the relevance of the normal variants that you get in the exam is that we need to make sure we don't confuse them with pathology. So this should not be confused with a fracture. This is just a persistent metopic suture. Great, now let's look at what's inside the calvaria. We'll start with the cerebrum first, then we'll talk about some white matter tracts, basal ganglia, and the cerebellum. The cerebrum is the largest part of the brain. It consists of the cerebral hemispheres, the cerebral cortex, and the underlying subcortical structures, and that includes the basal ganglia, white matter tracts, and the olfactory bulb. And it can be grossly divided into 5 lobes. So we have the frontal lobe anteriorly, parietal lobe, occipital lobe, and temporal lobe. And just deep to the temporal lobe, we have the insular cortex or insular lobe, which is seen on this diagram with the temporal lobe retracted down. And on these lobes, we have different sulci and gyri to increase the surface area of the brain, and we can identify them, and we can be asked about those in the exam. So we will need some landmarks, which I'll show you to help you identify which lobe you're looking at, which side you're looking at, and which suture or gyrus you're looking at. So the first and most obvious landmark you need to know is the interhemispheric fissure, which is this deep groove between the two hemispheres, separating them from each other, and that's where the false cerebri sits, highlighted in orange. So when looking at these axial and coronal views, always remember to specify right from left, not just for the exam, also when you're reporting later on. It's very important. You can lose lots of marks for not specifying right from left. So these are things to be aware of when you're answering your questions. All right, now let's start with the frontal lobe. So it has three surfaces. It has a lateral surface, a medial surface, and an inferior surface. The inferior surface is kind of low yield for the exam. I would recommend you read on it, of course, but I'm just going to cover the lateral and medial surface for now. And starting with the lateral surface, where we can see three sulci that will divide it into four gyri. So we have here the superior frontal sulcus, the inferior frontal sulcus, and the precentral sulcus, and that will give us the superior frontal gyrus, middle and inferior frontal gyri, and the precentral gyrus. Let's take a look at those on some images. So first we have a coronal MR, and we can see that we have an arrow pointing to a CSF space. So we know that this is CSF on this T1 image where the fat is very bright and the fluid is dark. So this is the right superior frontal sulcus. On this sagittal image, we have an arrow again pointing to this black line, which is a CSF space, it's a sulcus, and that is the inferior frontal sulcus. This intersects with another sulcus over here, forming like an L kind of shape, and that is the precentral sulcus. So you can see that they intersect together, and there's a gyrus here just below them, and that's the inferior frontal gyrus. I did not specify right from left because I cannot tell what we're looking at, whether it's the right side or the left side. So it's very important as well. If you cannot tell the laterality, do not add one just by guessing. You need to be accurate in your answers. This is an axial view where we can see the superior frontal gyrus again, intersecting with the precentral sulcus. And there's this important sulcus behind it, which we all need to recognize as well, which is our second landmark, and that's the central sulcus. This is important. It separates the parietal from the frontal lobes. So anteriorly, we have the primary motor cortex, or the precentral gyrus, and posterior to it, we have the primary somatosensory cortex, or the postcentral gyrus. And there are a few ways to help you identify that this is actually the central sulcus. So take a look here at the yellow dotted lines. That is the superior frontal sulcus. It intersects with the precentral sulcus, forming a T shape or an L shape. In this case, it's an inverted L. The sulcus posterior to them would be the central sulcus. Another way to tell is that the precentral gyrus bulges posteriorly into that sulcus. So it forms like a doorknob or an omega shape, inverted omega shape. So that's how you know that this is the central sulcus. Let's take a look at this axial CT slice. We have an arrow pointing to this CSF space. It's not pointing to the cortex or to the white matter, gray or white matter. It's pointing to this dark CSF space. And you can see that it's intersecting with another sulcus, forming a T shape. That is the right superior frontal sulcus. On the other side, the arrow is pointing to the gyrus. It's pointing to the brain parenchyma. So that is the left superior frontal gyrus. Here we have an arrow pointing to a CSF space that has this kind of omega shape. That is the left central sulcus. And the arrow here is pointing to the gyrus just posterior to it. So that would be the right postcentral gyrus. So the central sulci are highlighted here in yellow, and as a reminder, they look like an inverted omega. All right, let's move on now to the medial surface of the frontal lobe. There are two sulci here to identify. We have the cingulate sulcus, and we have the callosal sulcus just above the corpus callosum. And the cingulate sulcus will give off an ascending branch known as the paracentral sulcus, and a marginal branch known as the marginal sulcus. And this will divide the medial surface into four gyri. We have the cingulate gyrus, the medial frontal gyrus, the paracentral lobule. This is also part of the parietal lobe, so that's why it's a lobule, not a gyrus, and the gyrus rectus over here inferiorly. And on imaging, what do these look like? So Our arrow here is pointing to a CSF space again. This is the cingulate sulcus. Next, we have the ascending branch of it, so that is a paracentral sulcus. Next, this arrow is pointing to the second branch that comes off the cingulate sulcus, so that's the marginal sulcus. Just below the cingulate sulcus, we have the cingulate gyrus. Between the two branches, we have the paracentral lobule. Here we have the medial frontal gyrus, which is an extension of the superior frontal gyrus just medially in the interhemispheric space. And this would be the thin sulcus just above the corpus callosum, so that's the callosal sulcus. And finally, we have the gyrus rectus inferiorly here. All right, now let's move on now to the parietal lobe. The parietal lobe has two surfaces, a lateral and a medial surface. And we'll start with the lateral surface, where we can see two sulci. We can see the postcentral sulcus, and we can see the interparietal sulcus. And that will divide the lateral surface into three gyri: the postcentral gyrus, superior parietal lobule, and inferior parietal lobule. Let's see those on imaging again. So we have on the first image an arrow pointing to a gyrus. And as a reminder, this is intersecting with this sulcus. This is the inferior frontal sulcus with the precentral sulcus, which will make the one posterior to it the central sulcus. Therefore, this is posterior to it, and that is the postcentral gyrus. Now, this arrow is pointing to a CSF space, a sulcus posterior to the postcentral gyrus, and that is the postcentral sulcus, as the name implies. Here we have an arrow pointing to a gyrus, so that's superior parietal lobule. Now, let's take a look at the other image. We have an arrow pointing here to this gyrus, which is just anterior to this omega shape. That is the right precentral gyrus. The next arrow is pointing to the postcentral gyrus. That's the right postcentral gyrus. And this CSF space is the right postcentral sulcus. Great. Now we are onto the medial surface of the parietal lobe. There's one sulcus, which we've already seen, the marginal sulcus, and there are two gyri. We have the paracentral lobule and the precuneus. On imaging, so we have the paracentral lobule over here. We have the marginal sulcus just posterior to it, and we have a gyrus over here that is just posterior to the marginal sulcus. That's a precuneus of the parietal lobe. Moving on to the occipital lobe, it has also two surfaces. However, the lateral surface has a very variable anatomy, which is probably less likely to come up in the exam. Therefore, I would suggest you read on it, be familiar with the anatomy of the lateral surface, but I'm just going to focus on the medial surface. You have the parieto-occipital sulcus over here. You have the calcarine sulcus, which are the two main sulci to know, and they intersect together forming a Y shape. And in between, you have the cuneus. So let's take a look at those on imaging. The first arrow is pointing to the parieto-occipital sulcus. The second arrow is pointing to the gyrus in between the two sulci, and that is the cuneus. And finally, we have the calcarine sulcus. And again, they form roughly a Y shape. Now, we have another landmark to know, and quite an important one, and that's Sylvian fissure, or the lateral sulcus. And this will separate the frontal and parietal lobes superiorly from the temporal lobe inferiorly. And you can see the insula just deep to it, or the insular cortex is just deep to it. And here you see branches of the MCA, so it's a very important space to look at. Which brings us to the temporal lobe. There are here two sulci that will split it into three gyri. So we have the superior and inferior temporal sulci, and that will divide it into the superior, middle, and inferior temporal gyri, which are shown here on the right side. And on the left, you have the two sulci. All right, I'll move on now to some white matter tracts. I'll talk about the corpus callosum. There are other white matter tracts to be aware of, but this is one of the most important ones and the largest ones, and it links the right and left cerebral hemispheres together. It is C-shaped and it has four parts. Let's take a look at those four parts on an MR image. So the first part over here is kind of beak-shaped. This is the most anterior part of it, and that's called the rostrum of the corpus callosum. it makes a bend at this point where the arrow is pointing, and that's the genu of the corpus callosum. Next part is the body, and the most posterior part is the splenium. So other white matter tracts include the fornix, anterior and posterior commissures. I would suggest you read on them, but for the sake of time, I will not elaborate on those. Now, moving on to the basal ganglia, these are actually gray matter nuclei deep within the white matter, and there are three to know about. We have the caudate nucleus, and on an axial view, we're only seeing the head of the caudate nucleus, so it's important to specify that. We have the lentiform nucleus, which is formed by the putamen and the globus pallidus. If you can't see the distinction between them, which on CT, for example, you won't be able to, you can just refer to this as the lentiform nucleus. And you have the thalamus posteriorly. So what does that look like on MR? So in red, we have the head of the caudate nucleus. Here in blue, we have the lentiform nucleus. And in purple, we have the thalamus. The globus pallidus is presumed to be there, but we can't see it distinctly. And in between them, you can see that there are some white matter tracts. I've taken this image from the radiologist who shares really great content for part one and for anatomy learning. And he's labeled some of the structures for you. So you can see that there is a white matter tract over here. That's the internal capsule. It has an anterior limb between the head of the caudate and the lentiform. It has a genu where it bends and a posterior limb. And just lateral to the lentiform nucleus, we have the external capsule. So these are also structures to be aware of, and you could be asked to label these structures as well. All right, we've seen these parts. Let's take a look at this coronal image. We have here the right head of the caudate nucleus. On this image here, we have another gray matter structure, and that is the right putamen. You can see a separate structure from it, so we can be specific here. And on the left, it's pointed out that's the left globus pallidus, and this is the external capsule more laterally. And here we have the internal capsule, but the arrow is pointing to its genu over here. All right, and now briefly, let's talk about the brainstem. The brainstem is composed of three parts: the midbrain, pons, and medulla. We're starting with the midbrain. The most posterior part over here is called the tectum, or the quadrigeminal plate, tectal plate, which is composed of superior and inferior colliculi. We'll see these in a minute. We have the tegmentum in the middle here, which contains the brainstem nuclei, and anteriorly we have the cerebral peduncles. On imaging, we have anteriorly, we're pointing to the right cerebral peduncle. Excuse me. More posteriorly, we're pointing to the tegmentum of the midbrain. And most posteriorly, we have the quadrigeminal plate. And it's important to remember that the tegmentum is continuous between the midbrain, pons, and medulla. So we need to specify which tegmentum we're talking about. Sorry, I'm dying for some reason. We have an arrow here pointing to the quadrigeminal plate. And if we move more to the side, we can see what it's actually composed of, which is the superior and inferior colliculus. This part over here would be the midbrain tegmentum. Now, looking at the pons and medulla, they both have a ventral part, an anterior part, and a more dorsal part, which is the tegmentum. And on imaging, we can see here, this is the pons. We have the basilar artery just anterior to it. We're still seeing a lot of the temporal lobes, so we are not low enough to be at the level of the medulla. So that is the ventral pons. This is the tegmentum of the pons. And just below it, we have the medulla oblongata. The last part before I hand over to Alicia is the cerebellum, which sits at the base of the brain, and it has two hemispheres joined by a vermis. And it can be divided into an anterior lobe, posterior lobe, and a flocculonodular lobe. It is joined to the brain stem. excuse me. It is joined to the brainstem by three peduncles. We have the superior cerebellar peduncle joining to the midbrain, the middle peduncle joining to the pons, and the inferior cerebellar peduncle to the medulla. Let's take a look at some images. So we have here the foculonodular lobe on the left side. Here we have the anterior lobe, and here we have the posterior lobe. On CT, there's another structure that we need to look at. We can see this on MR as well. This is the cerebral tonsil. And in the midline over here, we have the vermis. Alicia, over to you.
All right, thank you, Harut. Hello, everyone. I'm Alicia. Thanks for joining us today. So I'm going to take you through the ventricles and the basal cisterns now. So we're going to start all the way back from the beginning and talk about cerebrospinal fluid. What is it? Where does it come from? And where does it go? So cerebrospinal fluid is responsible for providing protection to the brain and spinal cord. It's an ultrafiltrate of plasma contained within the ventricles and subarachnoid spaces. And actually, we only have an estimated volume of 150 mils of cerebrospinal fluid within these spaces, which probably doesn't sound like a lot. So here we can see, oh, just move forward my slide. So here we can see a diagrammatic representation of the ventricular system in the sagittal view. And within the three ventricles, there are these invaginations of pia matter, which are lined by modified ependymal cells and capillaries. So these are choroid plexuses, and they produce CSF. And you can see these as these red structures that we have here lining the ventricles. And this is another diagrammatic representation, but this time in the coronal view. And we can see our ventricles here. So we'll start off with these paired C-shaped lateral ventricles. And on either side, we have the interventricular foramen, or the foramen of Monro, which connect our lateral ventricles to our third ventricle, this midline structure here between the right and left diencephalon. We then have here the cerebral aqueduct or aqueductus sylvius, which connects the third ventricle to the fourth ventricle. And we can see here these lateral apertures. So they are known as the foramen of Luschka, and they link the fourth ventricle to the cerebellopontine cistern. Along with the median aperture, which we'll see in a bit later, these are three sites that the CSF can leave the fourth ventricle and enter the subarachnoid space. So here we have these two sagittal MRI images, and you can see on the left here, this is a T1 sequence. Fat is bright and fluid is dark. On the right, we have a T2 sequence. Fat and fluid is bright, so we can see our CSF as white. And we'll have a look again at these structures. So we have the lateral ventricle here, coming around to the third ventricle through the foramen of Monro. And this passes through here, the aqueductus sylvius or cerebral aqueduct into the fourth ventricle. Once the CSF leaves the fourth ventricle to enter the subarachnoid spaces through the foramen of Luschka and the foramen of Magendie, it will flow all around the outside of the brain and the spinal cord until it comes to these arachnoid granulations here that you can see. And these filter the CSF from the subarachnoid space into the dural venous sinuses. All right, let's take a look at all these structures again, but in a bit more detail, and we'll have a look at what they look like on CT axial image. So this is the lateral ventricle that we're seeing here. We'll start off here with this anterior horn of the lateral ventricle, which sits in the frontal lobe. We'll come around. This is our body of the lateral ventricle, which passes through the frontal lobe into the parietal lobe. Here we have the temporal horn of the lateral ventricle, or inferior horn, and as you guessed it, that sits in the temporal lobe. And here is our occipital horn, which projects back into the occipital lobe. This area here is called our atrium or trigone, and it's the area at the confluence of the occipital and temporal horns. So this is a CT axial image that we're now going to go through, and we can see these paired structures here. So this is the body of the lateral ventricle that we can see on either side. It's separated by this thin membrane, the septum pellucidum. And as we go a slice lower, we can now see we've got these two frontal horns projecting into the frontal lobe. And this is our trigone or atrium. And you can actually see these structures here. These are our choroid plexus, which might be calcified in some images that you might have noticed. Here we have our temporal horn, and here is our occipital horn. And they might appear quite slim, but this is how we would expect them to look in a normal adult brain. This is our third ventricle now. And we're looking at this on an MRI coronal image. So as I said earlier, midline thin structure below our lateral ventricles. So what does this look like on axial CT? So if you remember earlier, we talked about these frontal horns of the lateral ventricle, and you can see this passageway here. This is our interventricular foramen on either side, connects our lateral ventricle to the third ventricle here. We're seeing just the anterior part of the third ventricle and the posterior part of the third ventricle because they are divided by this interthalamic adhesions. But as we go lower down again, we can see the third ventricle here as a whole, and it's flanked either side by our two thalami. And as we go lower down again, we can now see this small dot here of CSF. This is our cerebral aqueduct or aqueductus sylvius. It sits in our midbrain and it's going to connect our third ventricle to our fourth ventricle, which is just here between our pons and our cerebellum. Here is our lateral aperture that we talked about earlier, or foramen of Luschka, one of the ways that the CSF will leave the fourth ventricle. And remember L for lateral, L for Luschka. And here is our median aperture or foramen of Magendie, the other way that the CSF leaves the fourth ventricle into the subarachnoid spaces. And once again, easy to remember because M for median, M for Magendie. Anatomical variants that you need to be aware of for the exam. So we have the cavum septum pellucidum, which is a normal variant where the CSF, where there is a CSF space between the leaflets of the septum pellucidum, as you can see here, looks a little bit like a butterfly. And the posterior extension of this space is called the cavum virgae. So when we see these two together, we would call this cavum septum pellucidum and virgae. I'm now going to go through the subarachnoid spaces and the cisterns. So cisterns in Latin means box. These are not ventricles, but they are compartments within the subarachnoid space where the pia mater and the arachnoid mater are not in close approximation. So they form CSF pools. And the flow of CSF through all of these systems are continuous, but we divide them into areas so we know what we're talking about. And as you can see, the names are pretty self-explanatory. So we're going to go through them and you'll see that they all sort of make sense. So we'll start off here with this pink structure, and you can see that's running along the corpus callosum. So that's our pericallosal cistern. You can see here on this T2 sequence, we've got this thin membrane. That's our lamina terminalis, sitting anterior to the third ventricle. And just in front of that, this dark blue space represents the cistern of the lamina terminalis. Here again, I'm going to point you to this is our optic chiasm. This is our pituitary infundibulum coming down to here, our pituitary gland. So the cistern in front of these structures is our chiasmatic cistern. So chiasmatic for optic chiasm, or it's also called the suprasellar cistern, supra above, cella above the sella turcica. All right, and here in light blue, just in front of the midbrain, in between the cerebral peduncles, is our interpeduncular cistern. The CSF then wraps around the midbrain in the ambient cistern, which I'll show you later, to reach behind in this orange area. This is our quadrigeminal cistern. Sitting just behind here is the quadrigeminal plate that Harut mentioned earlier. And behind our quadrigeminal cistern is this dark green area. This is our suprasellar cistern, as it sits above the cerebellum. Here in orange, as we go lower down, this is in front of the pons, so we call it prepontine cistern. Here in purple, it's in front of the medulla, or rather anterior to it. So this is our premedullary cistern. And finally here in red, the largest cistern is our cisterna magna, running through the foramen magnum. So this is an axial image that we can see. The CSF is bright. It's a T2 sequence. Just want to show you here. This is our suprasellar or chiasmic cistern that we talked about earlier. And on axial image, it has this characteristic star shape that you can use to remember it by. This is our midbrain with our two cerebral peduncles. So in between is our interpeduncular cistern. Going around the midbrain is our ambient cistern, and behind is our quadrigeminal cistern. I'm going to take a 5-minute break, you'll be relieved to hear, and then we're going to come back to the second half of this session.